Okay, so this section is about enzymes, section 6.4. And enzymes are a special type of protein that speed up chemical reactions. So they essentially act as a catalyst. And a lot of times you'll see enzymes names end with the words ASE. So if you see something end with the word or the letters ASE, ACE, it usually means it's an enzyme. So for example, the breaking down of sucrose, table sugar, happens because of the enzyme sucrase, and that's the enzyme that helps break it down. So this is an example of sucrose and water, and when sucrase is added, it helps break sucrose down into glucose and fructose. It's two monomers. Basically, the reason why enzymes are necessary is because every chemical reaction involves the breaking and forming of bonds. And if you think of it kind of like a hill, you have to hit a certain point in order to start a chemical reaction. You have to have a certain amount of energy. And that amount of energy that's required to start the reaction is called the activation energy. This often is either produced in the form of heat or, and when you heat something up, you can speed up a chemical reaction and give it enough energy. But sometimes heat won't do it. So when you're looking at the chemical reaction that you need to occur, you may have some reactants and some products, but you're going to get a certain amount of energy. And when you get to this tipping point to get it over the hill, the reaction is going to proceed and you're going to have your chemical reaction with your products. And generally, the products are going to have lower energy than what you started with. But if you can't rely on heat or you want to rely on something else, organisms especially will use catalysis to speed up their reactions. And this is the case where an enzyme can speed this up by lowering the activation energy without being consumed. So enzymes don't change the free energy, which we'll talk about later in this unit, but instead they speed up reactions that would occur eventually. So it's kind of like if you had a hill and you were trying to get a boulder over a hill, eventually through like some miracle, the boulder would get over the hill, like maybe it would be blown by the wind or something like that. But if you had someone pushing the boulder, that person would be kind of like the enzyme, and that really decreases the amount of energy you need to get that boulder up the hill. It's going to get up the hill and over a lot faster. So basically this kind of shows you the reactions would occur naturally without the enzyme once they got enough energy, but this is about how much time it takes, the x-axis, and you can see it gets to that activation energy a lot sooner when you have the enzyme, and it happens a lot quicker. Enzymes are very specific for the reactions they catalyze, and so when an enzyme interacts with a reactant, that reactant is called the substrate. And basically the way enzymes work is they are very specifically shaped so that they can bind to a specific substrate and form what's known as an enzyme substrate complex. The active site is the region on the enzyme where the substrate binds. So in a moment I'll show you a picture. But we need the enzymes to be specific and have a complementary fit, kind of like a best friends forever necklace, so that the reaction will work. And this brings the correct chemical groups of the active site together. Enzymes will change their shape when they interact with the substrate, and sometimes that affects the progress of the reaction as well. So you can see here we have the enzyme, and this substrate is perfectly made to fit right into this active site right here. And then when they come together, we call that the enzyme substrate complex. In a reaction with an enzyme, the substrate, like I said, binds to the active site. And the active site helps lower this activation energy barrier. So you can see here we have two different substrates. And the active site is right here. The substrates are going to come in and perfectly align to that active site, and they're trying to get into the right location. Now the substrates can go through the chemical reaction because we formed the enzyme substrate complex, and the chemical reaction can proceed, and the products get released. And then the active site is now available for new substrates. This is our enzyme. If we increase the substrate concentration, so say we put more of the substrates here, the reaction can be sped up even faster because if we have more substrates, they can bind to more enzymes and more reactions can take place. So if, though, all the enzyme molecules in the solution are bonded with a substrate, like you put too much substrate in, then the enzyme becomes saturated. There's no more enzymes that can take substrates. And at that point, the reaction can only be increased if we add more enzymes. So it'll kind of, if we add more substrate, it's going to increase, and then it'll eventually level off. One other thing that's important to note is that the conditions of a particular environment can affect the activity of an enzyme as well. So in addition to adding more enzyme or more substrate, when you change the environmental conditions, those can also affect the enzyme's activity. So temperature and pH can have a big effect on enzymes, and so can different chemicals. So each enzyme has an optimal temperature and pH at which they work the best. And if you think about, for example, enzymes in the stomach where it's very acidic, those enzymes are going to work better at maybe a more acidic, lower pH. And then there's other enzymes in your body that work best at neutral, around 6 to 7 pH, because a lot of your body is neutral pH. So, for example, in temperature-wise, typical human temperature or body temperature is 37 degrees Celsius. So you'll see that the enzymes in humans have the greatest rate of reaction around that temperature. But in heat-loving bacteria, their optimal temperature is a lot higher because those bacteria live in an environment where it gets very hot. Similarly, in the stomach, the pH is very low for those enzymes because those acids in the stomach that help break down and digest your food cause the stomach to be a very low acidic environment, low pH, and so those enzymes are going to work better at a low pH. But trypsin, found in the intestine, works a little bit better at a neutral pH because in the intestine, the pH is a lot more neutral. Cofactors are non-protein enzyme helpers. So these are other factors in the environment that can help an enzyme that are not proteins. They could be a metal ion, such as sodium, or they could be organic and have carbon. And so if it's an organic cofactor, we call it a coenzyme. A lot of these coenzymes are vitamins, or they help make, vitamins help make coenzymes. And then when we talk about enzymes not working, we have a few different types of inhibitions that can happen with enzymes. There's really two types of enzyme inhibitors. The first are competitive inhibitors, and these are inhibitors that actually bind to the active site of an enzyme. And so they're called competitive inhibitors because these are competing with the substrate. They're taking the spot of that substrate. Then we have non-competitive inhibitors, and these might bind to another part of the enzyme, and that would cause the enzyme to change shape, and then the active site may not work as well or at all. So some inhibitors include toxins, poisons, pesticides, and antibiotics. So here's an example of competitive inhibition. So normally, like we said, the active site is perfectly to fit to the substrate, and it's like a lock and a key. They fit perfectly. The reaction goes forward. In competitive inhibition, the competitive inhibitor binds to the active site and can also fit, at least in part of it, but now the substrate can no longer come in, and so it's competing with each other. A non-competitive inhibitor over here attaches in another part that's not the active site, but you can see it now changes the shape of the active site in such a way that it's not as effective. It doesn't work as well. You can see it doesn't quite fit the lock and key right here. But it's not competing with the substrate, it just changed the shape. In general, most enzymes are proteins, and if there is a change or mutation in the genes that make up that protein, then that enzyme may have a different amino acid composition and no longer work. If that amino acid composition is different, that could mean that the enzyme doesn't work as well, or the substrate can't bind as well. And so the way new enzymes have evolved have been in cases where maybe that change or that mutation in shape or substrate specificity is favored, and then by survival of the fittest and natural selection, that enzyme might have been selected for and those genes passed on. So we'll end there for 6.4.